So I'm very pleased to welcome Dr. Magdalena Janecka, who's interested in coming to Columbia, we hope. So she's currently an assistant professor in the Department of Psychiatry and Genetic and Genomic Sciences at the Icahn School of Medicine at Mount Sinai. She completed her PhD at the Institute of Psychiatry, King's College London. After her first postdoctoral position at the University of Oxford, she joined the SIVA Autism Center at Mount Sinai in 2017. Dr. Janecka's research leverages health-based registries, genetic data, and epidemiological approaches to better understand the mechanisms underlying the observational associations between perinatal exposures and neurodevelopmental disorders. She has a very specific interest in autism and has spoken to the psychiatric epidemiology group before, and very excited to hear your talk, and so welcome.
Thank you so much, Louise, for this really kind introduction. Those of you who are on Zoom, please let me know if you can't hear me. I'll try to make sure everything gets picked up by the mic, but give me a shout if you're having any issues.
We can hear
you. So yeah, it's of course a great pleasure to be here, and I really look forward to sharing with you some of my lab's work on the role of environment in autism etiology, and going over some big picture questions and challenges in the field as I go along. I hope the talk will stimulate a nice discussion. In case you have any follow-up questions or anything comes up later on, please feel free to reach out to me at any point. And it's normally already at this point where I disclose that we actually don't know that much about the role of environment in autism yet, but I'll be trying to convince you why it's both really important and really difficult to get this right. So I start from making the case for the importance of better understanding the role of environment in autism. I'll explain some of the ongoing challenges in the field and how my lab has been tackling these questions for the last 4 years. Before I move on, I need to acknowledge everything we do is teamwork. I will do my best to acknowledge individual team members as I go along, but I both won't be able to cover everything these guys do, and also want to acknowledge upfront that these guys did all the heavy lifting behind the analysis we'll be seeing today. So I believe many of you will be well familiar with autism, but just to make sure we're all on the same page, autism is broadly considered to be a neurodevelopmental disorder with early, most likely prenatal origins, where something in the brain development goes out of its typical tracks. And in approximately 1.5 to 2% of children, this results in a diagnosis of autism, which manifests itself as deficits in social communication and restrictive repetitive behaviors. Severity of the symptoms will vary vastly between affected individuals and can fluctuate across the lifetime, but autism is broadly considered to be a neurodevelopmental disorder with currently no cure. And approximately 4 times as many males as females are diagnosed, and there is a broad consensus that this imbalance arises partly due to the diagnostic criteria being slightly better tailored to detecting autism in males, as well as females being better able to hide low level of autistic traits, something that in the literature is referred to as camouflaging. But most likely true biological differences contributed as well. These differences are broadly referred to as female protective effect. No one really knows what this female protective effect really is, but throughout the literature, females are shown to have a higher liability threshold, meaning they need higher scores on standardized assessments, they need higher genetic liability, and often presence of additional cognitive impairments to receive a diagnosis. And critically for my line of research, autism is considered to be a complex disorder with both environmental and genetic factors contributing. The relative importance of these two is still a subject of a debate, and approximately for 20% of the cases, there exists a single genetic cause that we can identify, And these cases are referred to as syndromic. And examples of these syndromes include Fragile X or tuberous sclerosis. So when I'll be talking about etiology of autism for the remainder of my talk, I'll be focusing on the remaining 80% of so-called idiopathic autism, a disorder with no clear cause. So starting a little to tell you a little about the genetic etiology of autism, thanks to some seminal studies by Susan Falstein and Mike Ratter, drawing from a much higher concordance in autism diagnosis between mono compared to dizygotic twins. We've known for decades now about the prominent role of the genetic component in autism, although at the time, and that's a quote, what is inherited was unknown. And skipping over decades of discovery, by 2023, thanks to efforts of large international consortia, many of the genetic factors have been identified, and also increasing the sample size from these 21 twin pairs and Falstein and Ratter study to millions of individuals from the Nordic countries linked through complex familial relationships, work done by my colleagues from the Sievers and Sven Sandin and Avi Reisenberg, estimated the heritability of autism to be approximately 80%, adding to a firm body of evidence that autism is of predominantly genetic origins, which exposes an elephant in the room and likely makes us pause and ask ourselves a question: why do we even bother studying environment in the context of a disorder that is so overwhelmingly heritable? And there are multiple answers to these questions, but I'll start from a real world one. I came to autism research from like vast experience volunteering with families of affected individuals. And I know, and everyone who's interacted with these families will know about the huge extent of guilt that exists among these families, specifically mothers, who often blame themselves for specific pregnancy or like other early life factors that could have brought about their child's condition. And I think to the extent that certain environmental factors could be causally associated with autism, it's critical, we know it, but if they don't, it's just as important to get the information out there. And we need robust and rigorous research to accurately accounting for confounding, to equip parents with accurate and reliable information. So I think that in itself should suffice, but there is also a more nuanced scientific answer. And to get to it, I think it's worth spending a bit of time discussing what the heritability measures of disease actually tell us. So heritability, this 80% in the context of autism, informs us about the variance in the phenotype attributable to genetic variation in a given time and place. And this very narrow and very specific definition often gets misrepresented and misunderstood. And the key caveat in inferring that genetic factors alone contribute to 80% of autism etiology is the non-independence of genetic and environmental factors, which gets ignored in these models. So the first issue here are the gene-environment correlations, whereby certain environmental exposures are genetically determined. So to give you an example, we know that smokers that smoking causes cancer, but we also know that propensity to smoke is under genetic influences, which already gives us a feeling that deciding whether a trait is genetic or environmental is going to be difficult. And in standard heritability models, the variance in the phenotype gets partitioned between effects of genetic and separate effects of genetic and environmental variation. But to the extent that these two are correlated, not accounting for it, will end up increasing the variance we attribute to the genetic effects and inflate the heritability estimate. And a similar inflation of the heritability estimate will happen due to gene-environment interactions, whereby certain genetic variants make us more susceptible to the effects of the environment. And again, simplifying and using lung cancer as an example, we can assume for now that like, neither genetic factors or smoking can cause lung cancer, which will only happen if the two act together. And to the extent that environmental factors run in families, which we know can happen for all sorts of non-genetic reasons, and we do not account for this gene-environment interaction, this component will again end up inflating the variance attributable to genetic effects, i.e. heritability. So heritability measures of disease are incredibly useful to the extent that they provide us with the upper bound of the effects of genetic factors on the trait. But it's important to remember that the estimates of heritability we get from these models will include both true effects of genetic variations, as well as effects of mediated for gene-environment correlations and interactions. And in autism, as well as in many other complex traits, we mostly just don't know how this estimated genetic component exerts its effects on disease risk, including through or in interaction with the environment. So I think the need to better understand what environment is actually doing there is clear, but turns out, as I highlighted at the start, it's actually a fairly difficult thing to do. And we're still a very long way from understanding of what happens at this interface of environment and brain development that can result in a higher risk of the disorder. And why is that? First short answer, which will come as no surprise in this department, is because confounding in these observational associations is pervasive. And as I said, like at the start, certain estimated genetic effects can occur through or in interaction with the environment. But to keep the argument balanced, it's fair to also acknowledge that many of the observational associations between environmental factors and disease arise due to background confounding. And to illustrate this notion, again, we can go back to our example of lung cancer. It's possible that smokers have a higher risk of lung cancer because smoking causes lung cancer. But it's also possible that certain genetic factors are independently associated with both smoking and risk of lung cancer. And that's a scenario whereby like that if the genetic factors are independently associated with exposure and outcome, we refer to this scenario as genetic confounding. And we can replace in this causal structure, we can replace exposure as some perinatal factors operating before the child is born, use autism as an outcome. And if I was to summarize my research in a single graph, it's probably that, with us trying to distinguish the causal effects of the environment from those that arise due to background confounding, genetic or otherwise. And to give you an example from one of our ongoing projects, we investigated whether genetic confounding could be in place for exposures like preterm birth and hypoxia, which are relatively common birth complications with a robust association with autism risk. And to investigate that, Vahe Kachadurian in my lab and I designed a study in the SPARK sample where we leverage the notion that genetic liability to disease is not a binary 0-1, but it's continuously distributed in the population. And then on top of that, we also made two assumptions that there's binary liability thresholds that underlies this binary case control distinction. And then we added two extra assumptions. First one was that this polygenic liability to autism will be higher in members of multiplex families, i.e. families with multiple affected individuals, where each family member is on average closer to the threshold and more of them end up crossing it to become a case. And we also assume that this polygenic liability would be higher in families with affected females, with females again having been shown to need higher genetic burden to receive a diagnosis. And the recurrence risk in siblings is higher if the proband is a female. So these were our assumptions. And when we stratified the SPARK data based on these family characteristics, we observed that preterm birth and hypoxia were 50 to 60% more common in members of multiplex families, unaffected members of multiplex families, and 20 to 40% more common in unaffected members of families that had a female as a proband compared to families with only affected males. And what these results show us is that the risk of preterm birth and birth hypoxia increases in individuals with higher estimated genetic liability to autism, which again is consistent. It's still something in preparation, but that's something that's consistent with potential genetic confounding in these observational associations we started with. And this really highlights that like examining confounding is warranted even for the associations that have been like fairly well established on the observational side. And the second answer to this question, like why is it so difficult? Why don't we have all the answers yet? And that's something related to the first one, is that environment turns out is fairly complex. And to illustrate that, I'll use an example of SSRIs, which are selective serotonin reuptake inhibitors, which is a class of antidepressants with a very robust association with autism risk. So maternal use in pregnancy is associated with offspring risk. But the association has also been subject to a long debate about the role of potential confounding by maternal indication, i.e. the extent to which the effects we observe arise due to the actual role of SSRIs rather than maternal depression or genetic factors operating upstream. And both our work and many other people's works show that this confounding likely is the case. But to complicate it even more, it turns out that depression itself is not an isolated medical risk factor. So this is a graph from a recent project done in the lab where we looked at the physical comorbidities in women who had a mental health diagnosis in pregnancy, which mostly included women with depression and anxiety. And turned out maternal mental health disorders in pregnancy were associated with a host of other physical problems, including higher risk of digestive, circulatory, and endocrine disorders, suggesting that some of the potential effects of depression could arise in part due to the correlated effects of physical disorders. And in fact, two very talented high school students who were in my lab summer 2021 were able to show exactly that the association between maternal mental health problems in pregnancy and adverse birth and pregnancy outcomes is strongly attenuated and becomes non-significant once we control for measures of physical health. So yeah, given pervasive confounding and the fuzzy definitions between what is environmental and what is genetic and the complex network of these different relationships, how do we move forward? And while these challenges to many people represent a nuisance, for us it's something that makes it fun and interesting and really drives the design of many of our studies. So for the remainder of my talk, I'll focus on some of the ongoing major NIH-funded ongoing projects in the lab. And these focus on the effects of maternal health in pregnancy and medication use on the risk of autism and other neurodevelopmental conditions. And the broad structure of these two projects is fairly similar and involves first comprehensive diagnosis and medication-wide searches, which are followed by a series of hypothesis-driven tests to better understand the source of the associations we observe in the first step. And I need to be completely transparent that the scope for inference in those two falls short of what we get from RCTs or other experimental designs. So even after we finish this project, there will be more work to do. But I believe that these steps are critical to first identify potentially novel exposures and then elucidate broad underlying mechanisms to nominate the exposures that warrant the subsequent investigation the most. In both of these projects, we try to triangulate evidence for the underlying familial confounding. Many of you will be familiar with this piece by George Davis Smith and Marcus Monafo from 2 years ago, which basically kind of argues, and I agree strongly, that every single scientific method, especially in the non-experimental space, will be associated with like a unique set of like strengths and limitations. And to arrive at the most robust conclusions, we really need to integrate evidence from across these different lines of investigations, i.e. triangulate. And so in line with that, in our first project, we start from screening 362 maternal diagnoses in pregnancy, which is what was left after the data quality control. And again, that's a study that we ran in a Danish birth cohort of over 1 million individuals. We account for data censoring by using Cox proportional hazard model, account for presence of siblings in the data set by calculating robust standard errors, and account for a number of covariates, mostly related to family socioeconomic status. We account, of course, for multiple testing by using false discovery rate. And after all these steps, we observed that 52 maternal diagnoses were significantly associated with autism risk. And here on this graph, each dot represents a unique association between one maternal diagnosis and autism risk. The higher the dot, the more significant the effect. And the larger it is, the higher the effect size. And the top line here represents the threshold for statistical significance after multiple testing correction. The dots are colored based on the broad ICD-10 categories that we use in the analysis. And as you see, some of them have been already grayed out. And those gray dots indicate diagnoses that were no longer significant after accounting for comorbidity between them by putting everything in a single model. And at this step, we end up with 30 maternal diagnoses significantly associated with autism risk. And those included mostly like well-known and well-established association between maternal psychiatric health, pregnancy and birth complications. But we also discovered few new associations, including breast cancer or maternal injury during pregnancy. And I must stress that across this plane, the mechanisms underlying those associations are likely very diverse. And that's something I'll get onto in the next couple of slides. Before I move on, I just want to introduce Candice, who was a master's in epidemiology student in my lab, who luckily decided to stay with us past her degree. And we're currently applying for a diversity supplement to explore this effect. So here everything's done in Denmark. We're going to explore this effect, how these effects differ across diverse racial, ethnic, and socioeconomic groups in a sample from Kaiser Permanente, Northern California. And so, kind of zooming on the mechanisms, many studies looking at these associations, sorry, I'll grab it. Many studies looking at these associations assume some extent of causal effects of maternal diagnosis in pregnancy, on some aspects of in utero environment that then contribute to increased risk of autism. And some of these factors can include, for example, maternal inflammation that arise downstream to maternal disorder. And of course, these effects are plausible given that placenta provides a robust shield, but many factors can still pass through. And the blood-brain barrier only develops gradually during prenatal environment. So it's perfectly plausible that factors from maternal circulation can cross the placenta and directly affect the fetal brain. So this is definitely a possibility, but Another one, oh yeah, and to the extent that these effects arise downstream to maternal diagnosis, they would differ between two siblings based on maternal health status during pregnancy. So that we will observe them for siblings whose moms were affected in pregnancy, but not the ones where mothers were healthy. So we refer to this as non-shared environment, as it could differ between two siblings born in the same family. But as I said, that's not the only potential mechanism. Genetic factors that contribute to maternal diagnosis could also be transmitted to the child and pleiotropically affect both the risk of maternal diagnosis and autism in the child. Let's say pleiotropically affect the risk of maternal asthma and autism in the child. And that's something because this effect would only occur to the extent that the child inherits the relevant portion of genetic variation. We refer to them as transmitted genetic effects. And because they would be transmitted on average 50% of the time, they could differ even between full siblings. But importantly, these genetic effects could operate even without the need of being transmitted through their effects on the in utero environment. So again, going back to the notion of pleiotropy, it's plausible that certain genetic factors that increase the risk of maternal disorder also contributes to other aspects of maternal physiology and regulate things like, for example, folate metabolism with knock-on effects on the in utero environment. And because this effect will operate irrespective of whether the child inherits that portion of genetic variation or not, and irrespective of whether mother has the diagnosis during the current pregnancy or not, we refer to them as non-transmitted genetic effects on the shared environment. We're getting there. And the same genetic factors that influence health during pregnancy can also influence health before pregnancy, with consequences on the genomic integrity of maternal germline. And I must say, because these effects are very stochastic, I do not believe that they would contribute to risk in any major way, but it's something that we definitely need to acknowledge and at the end of the day account for. And lastly, maternal health during and before pregnancy could be associated with aspects of shared environment like socioeconomic status, with consequences not only on the disease risk, but also likelihood of receiving a diagnosis. And helpfully, a lot of these effects, but not all of them, are also in operation symmetrically on the paternal side. So by starting from the assumption that only maternal health during the current pregnancy can directly affect the developing fetus, we can compare the risk associated with maternal diagnosis during and before pregnancy and maternal and paternal diagnosis to get better insights about the source of the observed effects. One thing that we need to acknowledge as well is the fact that maternal and paternal genetic factors will be correlated to an extent due to assortative mating, i.e. the fact that we do not choose our partners at random. But once we know about it and account for it, it doesn't complicate things too much. And this is just, I won't go into details here, but this is just an illustration that we can also use these familial relationships from more extended family pedigrees to get even better insights into the source of the observed effects. And here we're comparing the risk of autism among children of mother's sister and mother's brother. So I'll be happy to go into this. I don't want to swamp you with that. I'm happy to go into that during the Q&A later on. So knowing about all these potential pathways allows us to look at the original associations, elucidate those with evidence for familial confounding, and identify those that are most likely to be etiologically relevant in autism. So going back to this diagram, again, differentiate the diagnosis associated with autism due to, their causal effect on disorder as opposed to background confounding. And again, what we do, we compare the risk in siblings who are discordant for maternal exposure. So where mother had the diagnosis in one, but not the other pregnancy. We do paternal comparisons where we compare risk associated with maternal versus paternal diagnosis. We look at the effects of the timing of the exposure, where, for example, this is a graph for false labor, aka Braxton Hicks contractions. This is the estimate of the effects of false labor on autism risk during pregnancy. And this is the analogous estimate, but for the diagnosis that occurs 1 year, 2 years, and 3 years before the current pregnancy. And what we can see here is that even false labor 2 years before the current pregnancy is significantly associated with autism risk, even after accounting for the exposure status here. And this pattern really indicates some extent of familial confounding going on, because there are no plausible routes through which false labor 2 years ago would affect neurodevelopment of the current child. And this is really in contrast to what we see for preeclampsia or skull fracture, where only the exposure occurring during pregnancy is associated with an increased risk of autism in the child, but not the exposures occurring prior to pregnancy. We can then also use the Icahn comparison, which I showed you the schematic of and we'll be happy to discuss later on. And at the end, we'll also go to the Danish eyesight data, where we have access to the genetic information for not all, but like a subset of children in the Danish population. And we'll be able to estimate the polygenic liability to autism in those children and compare this risk between children whose mothers did and did not have the diagnosis in pregnancy. And again, observing significant differences here would indicate that maternal health status in pregnancy is somewhat related to genetic liability for autism, indicating potential background confounding. And what must be said is that each of these methods is associated with like distinct set of problems and strengths. So for example, the sibling and timing comparisons rely on quite precise ascertainment of the exposure timing, which we can't do very well for chronic diagnosis. And similarly, paternal comparison and cousin comparison do not work that well for sexually dimorphic conditions. The autism polygenic risk is getting there, but it's still not hugely powerful. But the whole idea is that each of these methods is good for something else, and putting them together, we can reach most robust generalizable conclusions. And that we've preprinted something from a smaller Danish study a couple of years ago. We're finalizing writing up the results from the full sample and in those top 3 comparisons at the moment. So you can look up the previous one, but an update is on its way very soon as well. And now I'll focus on, for the next couple of slides, on the other major project, which on the other hand focuses on the role of maternal medication. And even though this grant was only funded in September 2022, I want to illustrate that except for familiar relationships, we can also use some properties of the exposure itself for subsequent causal inference. So as you'll definitely be aware, several groups of medications taken by pregnant women have been associated with autism risk, including antidepressants and antiepileptics. However, as I highlighted earlier on, the role of the background confounding in these associations is not very well understood, and that includes confounding by genetic, medical, sociodemographic factors. And because most medications taken by pregnant women have never been studied in the context of neurodevelopment, we start from a comprehensive medication-wide screen of all of the associations of all medications in our Israeli population-based data with subsequent diagnosis of autism in the child. And we do it, we run the main effects model, but we also do it in relation to exposure timing, its total amount, and stratified by child sex, given evidence for higher vulnerability in boys. Because of the unique healthcare structure in Israel, we'll be able to run the primary analysis in the prescription data set. But because in Israel, even the over-the-counter medications are redeemed with the insurance card, which, by showing the insurance card, you end up getting a discount for the medication. Even the over-the-counter medication gets recorded. So we'll be able to analyze both prescribed and over-the-counter medications. And then, as I highlighted as a follow-up, we're interested in identifying potential confounding, and except for the familial designs I've discussed previously, we're also trying to kind of get a better insight into potential confounding by maternal indication. And for that, we're using the kind of target-based pharmacoepidemiology design, which I'll explain in detail in the next slide, as well as the discontinuation analysis, where we compare autism risk in children whose mothers stopped taking the medication early in pregnancy versus those who continued taking it into second and third trimester. And lastly, our goal will be to establish the extent to which these findings are generalizable and specific to ASD. So to establish generalizability, we'll compare the observed effects in samples from Sweden, Finland, and the US. And again, here we're working with Kaiser Permanente and Northern California data. And to establish specificity, we'll look at the extent to which the associations we observed for autism are also present for other neurodevelopmental and broader developmental outcomes in the child. And as I said, a lot of effort will go into trying to disentangle the role of potential confounding by maternal indication. And for that, we're working closely with our colleagues from pharmacological sciences to redefine the exposure. And rather than test the effects of specific medication on autism risk, look at the effects of the medication grouped by the target that they act on, and test the effects of this target-based group on autism risk. So this is a little schematic to illustrate that. We're collating the effects of different drugs that all act on the same target, but are prescribed for diverse conditions, as indicated here by these differently colored dots. And then we test whether this target-based exposure is associated with autism risk in the child. And to the extent that the association between the drug and autism would arise due to this pharmacological effect, to the extent that the association arises due to confounding by indication, this approach really just introduces more noise because we're grouping together medications prescribed for a diverse set of conditions. So we're less likely to identify an association compared to a situation where we have a single medication. And to the extent that the effect is mediated by the pharmacological interference with the target, we're really collating together multiple exposures with the same mechanism of action, which increases our power. And it must be said that this approach only works for drug targets that are fairly heterogeneous in terms of their indication. And for those, the approach will help the most. And these are the results from our primary analysis. So as you can see, the indication composition is fairly heterogeneous for many of the targets, but not all of them. And for ones like this, the method won't help at all. So for those ones that are not heterogeneous, we just need to acknowledge it up front and know that for those we'll need other evidence. Because here the exposure, the target and maternal exposures are fully overlapping. We can't say whether the observed effects arise due to one or the other. But to the extent that the method works, another strength is that through its focus on the biological properties of these medications, we're getting better insights into potential pharmacological targets that are etiologically relevant to autism. And the method is also more amenable to replication in other settings because the method relies less on the prescription patterns in any specific settings, because we're focusing on these functional consequences of the drugs. And I've done a smaller version of this study a couple of years ago. It was published in JAMA Psychiatry, and they also interviewed me for the podcast. So I never listened to it myself, but in case you'd like to find out more about the method and what we found back then, it's there. And now for the remainder of my talk, I'll focus on a project that I first thought was like relatively unrelated to everything I do, but like slowly dawned on me to be like more and more kind of like relevant and like fundamental to everything we do down the line. So Candice from one of the earlier slides, and I at the time was a high school student in my lab doing her high school bioinformatics placement. And both Aya and Candice were interested in racial disparity in diagnosis of multiple mental health conditions. And after brainstorming more, whether there is a way we can study this further, we realized that actually studying the concept of overdiagnosis is very difficult because it's close to using electronic health records, it's close to impossible to distinguish truly higher rates that arise due to higher levels of trauma or certain exposures in some communities from higher rates of diagnosis that arise due to diagnostic racial bias that occurs at the point of diagnosis. So only the second scenario represents overdiagnosis. So a situation where the diagnosis is given where it shouldn't be. And to study that, we decided to then simplify our question a bit and instead focus on looking at whether there is evidence that patients from different race, ethnic groups receive different pharmacological treatments for their conditions. And for that, we focused on schizophrenia, but to have two comparison groups, we requested data on all patients diagnosed with schizophrenia, depression, bipolar disorder, within Mount Sinai hospital system, and that included both demographic and prescription information. And we tested whether there is evidence that patients of certain race ethnicity groups have higher likelihood of receiving specific nervous system medications for their condition. We adjusted for age, sex, insurance status, comorbidity, BMI, and weight, and always included the location of the provider as the random effect in all the models to account for the fact that these prescription patterns could differ across different locations in the city. And what we observed, this is first for the broad groups of medications, where you have different race ethnicity groups on the x-axis and different groups of medications on the x. I don't know if you can see the numbers very well, but blue tiles indicate lower likelihood of receiving the group of medication compared to white patients, whom we always use as a reference group, assuming they receive standard treatment. And the red tiles indicate higher likelihood of receiving the medication. And only those with asterisks were statistically significant after multiple testing correction. So in this broad analysis, we only observed that patients who self-reported as Black African American or who did not report any race ethnicity were less likely to receive antidepressants. And then we looked at that at a higher resolution, i.e. resolution at the level of specific medication. And here for clarity, I'm only showing significant associations. And as you can see, this overall lower rates of antidepressants in Black African American individuals are replicated here at the level of individual medication. But we also observe that despite no overall differences in the rates of antipsychotic prescriptions, Black African American and Hispanic patients were more likely to receive risperidone and haloperidol, but less likely to receive clozapine. And then, as I said, we also looked at patients with depression and bipolar disorder to see to what extent these effects are specific to schizophrenia or generalized to other mental health conditions. And what we saw that was that these patients, here everything was significant, like the sample size was much larger for those diagnostic groups. Patients with depression and bipolar disorder were significantly less likely to receive antidepressants and mood stabilizers, which are the medications used to treat these conditions, but significantly more likely to receive antipsychotics. And this is after, just to remind you, controlling for comorbid schizophrenia diagnosis. And although discussing these results in details is really beyond the scope of my current talk, what I want to highlight is that, even though these huge health registers are an amazing asset in studying causes of disease, we need to bear in mind that from the outset, as we analyze the data, we're carrying all of the societal biases and healthcare inequalities into our analysis. So we need to be very careful in the way we interpret everything we see there. And with that, I hope I have managed to convince you that environment is worth studying. And this is probably best done without either dismissing its role due to high heritability estimates or inflating it due to these ubiquitous associations between ubiquitous observational associations. Having said that, even defining what is environmental is problematic, with increasing evidence that many of the environmental exposures actually have a genetic background. And further on, we're dealing with pervasive confounding and demographic and socioeconomic structuring the data that make the interpretation of the observational associations even more challenging. But I believe that there are many ways forward. I think with the latest large-scale phenotypic and genetic data, causal inference designs that are out there, and also the ones that are actively being developed, triangulating the evidence, and also a fair dose of skepticism to just keep probing more everything we're seeing. And with that, I'd like to end and of course thank my lab who contributed a lot to all of that, and collaborators at Mount Sinai and beyond that. And happy to take questions.
Do we have a way of seeing the Zoom people? We can go to stop share.
Did you find the chat?
I'm not getting the cursor to it. Can we see?
Yes.
Oh, there we go. Is there a way we can see who's online? We can see
Yes.
the people. There we go. Right. Can we put them so that they can put themselves on camera to speak?
Yeah, I believe they have the ability to.
So if we stop share, right?
There they are.
There we can see better. There we go.
Very good. Ezra has his hand up.
Right, so Ezra, please go ahead.
Hi, that was an outstanding talk, Magda.
Thank you.
I have just one question, which you could probably predict, which is people I know you and many people in the groups you work with are really focused on the polygenic risk score.
Yeah.
I feel that there's at least as much to be learned from the de novo mutations. And there seems to be this assumption that they're entirely random. There is a lot of random stochastic processing mutations, just like in everything. But there are also environmental factors that increase the risk of mutations, and just for example, radiation, low folate, whatever, there are many. And in some ways it's more tractable, to compare something that occurs, and whether these environmental factors are related to germline mutations, and in that, because it's more, the biological pathways are more easily understood with very large effects.
Yeah, I mean, I could not agree anymore. I think like often we look at common variation because it's easier, it's out there, and because of the kind of like overwhelming assumption that these effects act additively, and we can understand their effects on disease better, as opposed to rare mutations which are rare, which are more diverse, and which require different technology to detect. So I think a lot of, I think the focus on the common, aka polygenic kind of side of things mostly comes from, not dismissing the other side. It's the data that is out there. And I think I agree with you, we should be moving more to just, larger scale, like full genome sequencing studies that link the rare variation with environmental exposure. But I think at this point, we just like often do not have the data. But yeah, but another angle to that is that a lot of these rare mutations have been linked with complications of birth and pregnancy. That's something like I always felt like, why is no one studying the role of preterm in carriers? If we believe that preterm does something to the child, why do we dismiss it in those carriers of rare mutations? So I think there is a lot of kind of potential in integrating the evidence from both the rare variation and environmental factors and their joint consequences for disease risk.
Thanks, Magda. I'll let up.
Thank you so much, Ezra.
Ezra, while we have you on camera, can you hear me?
Yes.
Oh, brilliant. Good. I just wanted to make sure. I'll speak up just a little bit so Magda doesn't have to repeat my question then. So I was fascinated. So I thought your slides were wonderful, and the way you walked us through the studies was really clear.
Thank you.
And I enjoyed watching. It was a masterclass in that. I wrote a couple things down, so I'm trying to summarize. So I think some of the earlier work that you showed us was really trying to identify key confounders that might clarify the relationships of in either direction, whether clarify the gene outcome relationships or the environment outcome relationships, right? I think you talked about clarifying both of those. And then you started to talk about exposures, identifying maybe perhaps some of these confounders or other covariates might be warrant subsequent investigation, possibly with RCTs. So I wanted to hear a little bit more about that and what future directions you might be thinking of there. And then I also, with that in mind, you also had some wonderful stuff that you cited, Ruth Dave Smith's work and the triangulation papers. How do you anticipate integrating all the different evidence that exists now in that sort of triangulation work? I wanted to hear more about what you were doing in that triangulation work, not so much theoretically how it might work, and especially back to my original question, especially in the context that I suspect there are very few trials at all in here to draw from.
Exactly. So starting from the RCT question, that's one of the key motivations for starting with observational research is that for forever, including pregnant women in RCTs was not allowed. Right now, you can include pregnant women in RCTs, but I think there are no RCTs kind of, because it's not ethical to expose pregnant women to something that you think could potentially be harmful. So I think we need to, I think before we get there, we need way stronger evidence from observational studies. And I think we need to get into the role of the confounding way more. We need to follow up with animal models and see where that gets us. I think at this point, running RCTs would be challenging for multiple reasons. Going forward, of course, like the highest kind of quality of evidence will come from that. So at the end of the day, I think you just need to balance the risk and benefits. So I think, if the risk seems to be sufficiently high that it's like overwhelmingly would outweigh the benefit, that probably doesn't even necessitate an RCT. If you if you see that, like, just like telling pregnant women, stop taking this because the risk is so much higher compared to the potential benefit. It's a different situation where the risk and benefit are much closer together. And I guess in this second situation where, potentially, stopping certain medications in pregnancy or, other factors we can control in pregnancy would have like far smaller benefit.
Right, so that's partly what I was asking. I think some of these will never get to the
Yeah.
RCT for a lot of this.
Exactly.
And so, so I appreciate the concept of triangulation.
Yeah.
I think the related concept that might be interesting here is synthesis, literature synthesis of existing observational work as a related, it's not quite the triangulation that was discussed in those articles. It's more akin to meta-analysis or mega-analysis as opposed to the actual triangulation.
Yeah, with the caveat that, if every study, we put in our meta-analysis has the bias, then,
Right.
we're just like increasing it by putting it together. So I think it needs to be like, very thoughtful synthesis of evidence. Because as I said, even those well-established associations could reflect background confounding. We don't know, we're not saying yet, do they reflect it or not, but, the evidence so far we're getting seems to suggest that there is some, genetic link between things like preterm birth hypoxia and autism. It doesn't mean that preterm birth and hypoxia do not cause autism. They could be like on the causal pathway. We still need to understand these relationships better. But yeah.
Just a very quick comment on that. There is the in-between, the natural experiments, quasi-experiments, changes in the population, and even you can have a difference in differences. So that's kind of, stronger than an ordinary observational study, but not a randomized trial. And you can do that.
Well, I was going to ask, in that vein, I noticed there was a discontinuity analysis you did in one of the studies, which is, I think, speaking to Ezra's comment. Maybe you could talk a little bit more about that as a.
Yeah, so there are certain designs that like allow us to kind of disentangle the characteristics of, do people who are exposed and non-exposed, are they like systematically different in any way? And this is like, discontinuity analysis is one of the kind of designs that helps here. Because if we're assuming that all women taking the medication early on had the indication at some point, then only observing the effects only in those who continue taking the medication into the period of pregnancy where brain actually develops, because brain doesn't develop until like second trimester. So if someone stops taking the medication early on in pregnancy, we should not really see a huge effect on brain development down the line. And then comparing the groups of women who stop versus do not stop taking the medication really allows us to, dig deeper into that and see like, are these groups systematically different? And, do the effects we observe on the disease risk, do the effects we observe differ between these two? But yeah, I think like that's the beauty of, using like just like leveraging observational data rather than, necessarily running new kind of experimental studies. I think there is, a lot, as Ezra said, a lot to be done with those
There are design opportunities with observational data, not true experiments.
It's like, natural experiments
Right,
that you can, really use to,
And that might be part of the contribution to the larger triangulation that you're talking about. In the absence of actual RCTs, I think there's a lot of power to what you're suggesting.
Well, I think we're out of time, and this has really been fabulous. So, like all of us, to thank you for coming.
Thank you so much. Bye, Ezra.
Thank you.
Thank you.
Thanks for everyone who joined.
Thanks, Ezra. Thanks, everyone.